bioactive compounds that make them healthier. The genomics laboratory is a beautiful example of how technology has changed the face of science. So an example I like to use is the Human Genome Project that went on in the United States and elsewhere in the previous decades, where it took more than 10 years and hundreds of billions of dollars to generate the nucleic acids that are in each and every one of our cells, and it's also the blueprint in all plant cells and fungal cells. It is sort of the basic cornerstone of our genetics. So each one of us has DNA, for example, that is almost identical between individuals, but there are very subtle differences in the DNA that is in each one of us that makes us individuals different from each other. One of the things that we can do in the genomics laboratory is to do experiments to try to understand those subtle differences and to try to understand why some people are resistant to various diseases such as cardiovascular disease or cancer, whereas some of us may be more susceptible. So to try to understand that on the human level gives doctors more targets to be able to make people more healthy. We can use these same techniques to understand very subtle differences in plants, for example, why some plants are more drought resistant, or why they have better bioactive compounds that make them healthier. The genomics laboratory is a beautiful example of how technology has changed the face of science. So an example I like to use is the Human Genome Project that went on in the United States and elsewhere in the previous decades, where it took more than 10 years and hundreds of millions of dollars to generate the sequence, the actual complement of DNA in every cell in the human body. I like to use this as an example. So that technology that took place during that time was based on what is called a 96-well plate, so that 96 different experiments could be conducted at one time. And there were large, almost factories at many universities and genome centers across the country that ran these over and over again for decades. This particular fiber optic plate has 1.4 million wells on it. You can't even feel the wells because they're only 50 microns across. They're so small. And we can generate the same amount of information that took 12 years and hundreds of millions of dollars. We can now generate that information in 2 weeks for a few $1,000s.
That's what we need in Ecuador.
Point out one of the machines. So these various instruments here are what we use for what's called DNA sequencing. It's the instruments that are used that can generate enough data in a single tool of the information on a given individual. So if I were to give a blood sample and we put it through the series, then we could generate information about my specific DNA complement that would illuminate my susceptibility to disease. It's part of this revolution that is helping us work towards very customized, tailored medicine for each individual. So there will come a point in time in the near future when an individual will go to their physician with a particular ailment and they'll say, well, let's look at all of your DNA, and then that will help us tailor the right drug or the right medicine or the right approach to help you get better.
Mr. President, the other thing that this machine can do is from the plant side. So the human genome has been sequenced, but there's many plants have not. Only a few plants have been. And again, a lot of the nutritional compounds and interesting drugs and foods that will come out will come from plants. And with this suite of instruments, we can sequence the entire plant genome in a very short period of time. As a matter of fact, this lab did blueberry. We did the blueberry genome.
We need this in our country. We are the most compact, diverse country in the world.
Yeah, and the amount of the number of undiscovered genes and interesting biological things in those plants is really a fantastic resource. It's these machines that are the first step in finding those.
And why they're so good for you and to understand even the subtle differences between different strains that have perhaps more powerful antioxidants or bioactive compounds.
In this lab over here, we are trying to decide when we can find bits of the brain tube that flows into the blood. Instead of having to do surgery with blood, we can take a blood sample and try to find evidence that the person has brain tumor. And then the surgeon, then knowing that, will go in and do the surgery to put out the brain tumor. To do that, we require some very complicated instrumentation. So that's a really good example of a really unmet medical need. No one can fix this. The success rate for people who are very unfortunate with brain tumors is very low. And so it's a very compelling research project, because if we can fix it, it makes a huge difference to society, and particularly those people with a brain tumor. So that's one example where the technology is important, but the execution is more important.